they don't know that they might have sex.
They don't know that they have.
Yeah, well, they don't. It'd be great.
Oh, microphone. Sorry, microphone stuff.
Yeah, that's a good point.
I forgot about this.
Oh, you've already got.
I've got it, yeah.
You don't need one.
I don't need one.
No,
I can
I will project naturally.
There's
That's definitely the sound recorded. Good morning, everyone. Well done for making me here for 9 o'clock. I've been asked to make one local announcement, which is the end of conference or workshop evaluation forms. And it's been asked to make you aware that there are two. One is because EMBO, who have kindly provided funding, I assume, that's their main contribution, and bags, asked for an evaluation, and also the Newton Institute always asks for evaluations of everything that goes on here. If you could please do those, it's of value to everyone, even if it doesn't feel like it is. And I assume there's boxes or something to hand them in. On the conference desk.
Yeah.
That's that. And then we shall get going. It's my pleasure this morning to welcome Ingo Ebersberger from Vienna nowadays.
Right.
And the job here is always somewhat redundant because nowadays they always have the talk up there. So you can see how to spell his name if that was a problem for you.
I hope I didn't misspell it, so.
And clearly the talk will be on human genetic ancestry.
Thanks very much for the introduction. And first of all, let me just explain that I will present the work of a couple of people, total 5 people. Last but not least is Arndt von Häsler, which I think most of you would have expected to see instead of me. But unfortunately, he couldn't make it to that conference because of his kneecap, which is still kind of damaged. He broke it a couple of months ago, and he's busy with other things. So he asked me to act as his replacement, which I, of course, agreed on. And so I will present now our work on human genetic ancestry. And as an extension to this, I subtitled it When Branches Get Short, because the second part of my talk will no longer directly deal with human genetic ancestry. And last but not least, as an introduction is, so our acronym, which is CBIF, right? So one of the thousands of cryptic acronyms in the world of science is meant to say Center of Integrative Bioinformatics in Vienna, which to my opinion is as cryptic as CBIF. So who knows what integrative bioinformatics is? So let me first start by showing you a picture. I think it's Napoleon. And he's pointing towards the direction, and this might be the direction where we should go with evolutionary research in order to end up with the all-embracing answer to how evolution works. But this is actually not the reason why I show this picture, but rather it is a very detailed picture, right? You can see the color of the horse, you can see the hat, as you see it's a male, even we can think about that it's Napoleon. So we've got loads of information out of this picture, right, just by looking at it. And this is actually, I think, the way how we would like to have a picture of evolution, right? Just looking at it, and we have an all-embracing picture of evolution. That'd be really cool, but since we are kind of humble, we would never assume that we are able to draw a picture that is all-embracing and representing all tiny details of evolution, but rather it will miss some tiny but maybe important details. And for that reason, for every particular evolutionary scenario, we will have to repaint a nude picture, right? Which then maybe just shows a black horse or a person without head or a female sitting on a horse or whatever. Right? So just by now thinking about how many evolutionary scenarios there could be, we can get an imagination of how many pictures we should draw, which are, I think, quite a lot. So for that reason, some people like these kinds of pictures, which in principle show the very same thing, right? We have a horse, we have a person sitting on a horse, although it does not point towards a direction, which actually we don't care right at the moment. But still, we get loads of information out of this picture. And actually, many people like this kind of drawing more than the first one. So what we are trying to do in evolutionary research is try to find models which are as simple as possible, which then describe nonetheless in a more or less comprehensive way the way how we think that evolution works. And this is simply one a way how to depict it. And the reason why I show is that one of the very simple models of evolution is the coalescent model. Just to give you a short, brief recap on that, although I think that most of you are familiar with it, is that the coalescent model kind of models how genetic lineages evolve in a population. So what we have a number of constraints in that model. We assume that the population model, that the population has a constant side. We assume furthermore that each random generation chooses its parent at random from the previous generation. And by that we can trace pairs of lineages back to a coalescent event somewhere in their shared common ancestral population. So Kingman, 1982, developed a continuous model that allows to estimate times between the coalescent events. And what most of us know that the coalescent rate for any pair of genetic lineages is proportional to 1 over the effective population size in generations, or 1 over theta in terms of substitutions. So just to explain the drawing a little bit, in black bold we see the tree that represents the genealogy of the species, so the species trees. The tiny circles are the individuals in the population, and in order to depict it correctly that our population has a constant size over time, I have drawn the same number of circles in any branch of our species tree, and this is actually the reason why the symbols get a little bit smaller. Then we have genetic lineages represented by contemporary individuals over here in 3 populations, P1, P2, and P3. And these green genetic lineages we can then trace back in the evolutionary history to coalescent events where 2 genetic lineages find their most recent common ancestor. And this we can then do past these 2 time points, T1 and T2, which are actually the time points where the populations or the species split in evolutionary history. So what happens, of course, we can have coalescent events only between individuals of one population. So in our simple model, we do not allow population coalescent events between P1 and P2 genetic lineages before they enter the shared ancestor population. And by that, we can kind of make up this genealogy by following the green lines back until the most recent common ancestor of P1. And then we can follow back a single genetic lineage backwards into time into the population shared by populations P1 and P2 until we find down here the most recent common ancestor then of these 2 now polymorphic lineages in that ancestral population. And once we have found that this most recent common ancestor of P1 and P2, we again have a single genetic lineage that we can trace back into history until the shared ancestral population of all 3 contemporary populations, P1, P2, P3. Again, we have now the lineage coming back from P3. We have both lineages present as polymorphisms in that population until it again finds its most recent common ancestor of P1, P2, and P3. So this is the model I will refer to in the rest of my talk. So what people usually do by assuming that coalescent model, they go take sequence alignments from whatever sequence data they get, hopefully orthologous parts of the orthologous genes or sequences. They reconstruct the genealogy of these DNA sequences or protein sequences, ending up in that sequence tree. And then, by ignoring the population genetic effects in that whole process, they then infer that the species tree has somehow the same topology and also, in many cases, the same branch length as the sequence tree that we have reconstructed. And this is what we, or at least biologists, actually usually do. So this is also all very nice and very clean, as long as the basic assumption in this inference is met, that the population genetic processes act on a different time scale than the species processes. So the speciation process usually takes way more time than the coalescent process. in the population. And for that reason, that inference is in many cases a good approximation. However, what happens now when internal branches get short, right? Then we have a probability that the two genetic lineages descending from P1 and P2 do not find their most recent common ancestor within T1 and T2. Rather, they enter as separate lineages, the population shared by all three species. And now all of a sudden we have three genetic lineages in here. And there is no clear idea now, or no clear preference, which of these two genetic lineages will coalesce first. So any of the three possibilities are possible, and conditioned on the case that no coalescent event took place within T1 and T2. We have now the three equiprobable scenarios. We can have the case where P2 and P3 finds its partner first, spanning, and then later the lineage coming down from P1 joins in second, generating a sequence tree which is obviously incongruent to the species tree, right? So P2 and P3 appear as their most recent common ancestors, or their next close related populations or species. However, according to the species tree, they are not. The blue case is similar, but now P1 and P3 coalesce first, giving and again an incongruent sequence tree. And the third possibility is that, as in the topology of the species tree, P1 and P2 coalesce first, and then later P3 joins second. So this will give, of course, a congruent topology. However, recall that the most recent common ancestor of the genetic lineages of P1 and P2 are down here found only in the population shared by all three taxa. And according or conditioned on the event that all three lineages really made it until this ancestral population, we find all these three species equiprobable, so with probability 1/3. The most well-known example where this actually plays a role, this simple scenario, is the relationship of humans, chimpanzees, and gorillas, where we know that, or kind of evidence that this internal branch here is pretty sure. And this actually hindered the inference of the correct species tree for quite a number of times. I think it was several decades until eventually humans and chimpanzees were assigned as their closest relatives. So how comes? Or how can we nonetheless infer the correct species tree from the sequence tree? Simply since this branch has, of course, a length which is larger than 0 in this case. So we expect, in addition to the three ancient cases, which I explained in the previous slide, also to find that sequence tree where the coalescent event in fact occurred between T1 and T2. So in a total, we can then assume that the sequence topology, the sequence tree topology favoring the human-chimp grouping should be the most frequently observed in our data set. And this is actually what one sees when one look into DNA sequence data. And this is now a study we have done in our institute where we looked at a total of, I think, 11,500 genomic loci scattered throughout the genome, so randomly chosen, who are informative on the splitting of human chimps and gorillas. And what we see is that we encounter all three topologies, so human, chimp, and then gorilla grouping. Here we have the chimp-gorilla grouping and human is out group, and here as a third tree with the human with the gorilla grouping, and the chimpanzee is out group. And what we can see from this is that 9,147 out of 11,807 informative sequence trees, that is 77%, support the human-chimpanzee grouping, which is very nice and actually what one expects. So in principle, then we could say, yeah, well, the whole issue is kind of finished. We no longer need to consider working on that topic. However, there is some additional evidence which one should take into account. And this is the fossil record on primate evolution. And what people do? They wander around in the field and they dig out bones. And 5 examples of these bones I have depicted here with different timings. So Sivapithecus, for example, 30 million years before present. Egyptopithecus is the oldest example I will show, dated 33 million years before present. Sahelanthropus is the most recent one, 6.5 million years before present, proconsul 18, and Morosopithecus 21 million years before present. And of course, one can start to argue about these datings, right? Since they are kind of frequently used or treated as point estimates. However, there's certainly a range of variation in these datings. However, for the remaining part of my talk, I would simply assume that these datings are correct. Please keep that in mind. Now, what people do? They now analyze these fossils and screen for apomorphic characters, so evolutionary derived characters that are displayed in these fossils and which are also seen in all or a subset of the contemporary species as we see them nowadays. And by that, they can place these fossils onto the species tree. And this is what actually people have been done. So Egyptopithecus, for example, shows derived morphological characters that are shared by rhesus and the great apes, and for that reason should be placed here as a root. Morosopithecus, however, displays apomorphies nowadays seen only in the great apes as proconsul, and since they are dated at different times, both land on that branch. However, proconsul lived more recently than Morosopithecus. Sivapithecus, for example, is said to display apomorphies that we nowadays see only in orang, and for that reason, people place it on the orang branch. And Sahelanthropus is actually kind of an interesting fossil, or most interesting fossil from my perspective, because it shows morphological features which are nowadays seen only in humans. And for that reason, it has been placed on the human branch after the split of humans and chimpanzees. Now, why are these fossils interesting for us? Because they give us an idea when these speciation events might have occurred, right? Usually by giving a lower and an upper bound. So Sahelanthropus, for example, is treated as kind of a lower bound of the split of humans and chimpanzees. And by looking at the date, we now have to assume that humans and chimps split somewhere between more recent than later than 6.5 million years ago. So something like 7 maybe. Sivapithecus, for example, assumes or proposes that the orang lineage split something between 13 to 18 million years ago, and Morosopithecus and Egyptopithecus gives a lower bound and upper bound for the split of the rhesus. Now this is everything. If this would be true like it is, then we should be able to use any of these fossils as possible calibration points for our molecular clock and date the respective other speciation events. And this is a figure I stole from Patterson from the Nature paper 2006. It's exactly what they did. So they used 3 different calibration points, fossil calibration points, and dated and set the split, the speciation event these fossils are informative on as fixed, shown here in bold. And then based on that calibration point, they inferred the remaining speciation events, either further backwards in time or more recent. And if everything would be nice and clean, and if the fossil record would kind of agree in itself, then we would expect no matter what fossil we use, we would always end with the same timing. And what that table actually shows that there is somehow of an incongruency. So when we, for example, use Sahelanthropus, was the fossil eye that was placed on the human lineage, as a lower limit of the human-chimp split, and arbitrarily set what they have done, the upper limit to 10 million years ago, then we end up with a human-chimp split around 6.5 to 10 million years ago. And when you use this date as a calibration, then the gorilla would have split 7.3 to 19 million years ago, the orang 20 to 45 million years ago, and the rhesus 36 to 79 million years ago. However, when you now go to the human orang split, which people prior to detection of Sahelanthropus usually refer to as a calibration point, we end up with totally different timings. So we have the split set to 1318 to 18 million years ago of the orang. Based on that evidence, we would assume that humans and chimps split 2.9 to 6.3 million years ago, so way more recent than this one would explain. Gorillas, somewhere between 3.4 and 11.3 million years ago, and rhesus, 21 to 34. And the very same, or more or less the same results we get when we use Marathopithecus and Egyptopithecus as calibration points. So there seems to be some problems. These fossils are somehow congruent in themselves, and Sahelanthropus is kind of problematic. It somehow does not fit into the picture. And now, of course, we need to explain or find an explanation why this could be the case. Of course, one argument would be, well, they just misstated it, which I said I will not touch right at the moment. The answer actually is given by, or a possible answer, a very intriguing answer, is actually given by the coalescent process, which I've shown you before. So again, you remember that slide. We have three sequence tree topologies conditioned that occur with equal probabilities, conditioned on the case that we observe no coalescent event between T1 and T2, so in the ancestral population shared only between humans and chimpanzees. Now, by looking at our sequence data, we find the green case where chimpanzees and gorilla group together in 1,369 cases, so 11.5% of our sequence data, of the informative sequence data. And the blue case, where humans group together with gorilla, we find in the very same fraction, so in 11.4% of our sequence data. So for these two sequence trees, we have strong evidence that the most recent common ancestor, the human genetic lineage, shares with either chimpanzee or gorilla, dates back to the ancestral population shared by all three species, right? And now how about the ancient trees that are congruent to the species tree? By means of our method, of course, we are not able to detect them. However, we kind of assume simply by the model that in the fraction of congruent sequence trees, so the 9,000 whatsoever we had in our data set, there must be an unknown fraction of also ancient sequence trees, right? And simply by the model, we can assume that this ancient sequence tree occurs with the same frequency than the sequence tree favoring the chimp-gorilla grouping and the human-gorilla grouping, respectively. By that, we infer it to be also around 11.5%. So this now argues that a total of 35% of the human genome is kind of evolutionary old, which means that it finds its most recent common ancestor shared with either chimpanzees or gorillas, or their shared most recent common ancestor, only prior to the gorilla split. Now, how are these regions distributed across the genome, right? They are actually, when we plot our sequence data, shown here are chromosome 1 and chromosome 2, we find them pretty much everywhere. There are apparently some regions in the genome that appear to be free of these incongruent sequence trees, which is interesting, but I think, or at least from looking at the picture, they are pretty rare. But more interestingly is what happens when we now look into regions covered by genes or exons. And there we actually see that incongruent sequence trees occur with pretty much the same frequency, although a bit reduced, as when we simply use all the sequence data. So no matter where we look into the human genome, we find these incongruent sequence trees with pretty much the same frequency. And this is now an example when we zoom in into a region of human chromosome 19 and now actually map the genes in that region. We see that, for example, in this case, two genes are pretty interesting. This one would be gene 3, which over its entire length kind of favors the grouping of chimpanzees and gorillas, whereas for gene 4, which would be lilRab B, has kind of a mixture between these two different sequence trees. And in fact, when we now draw a maximum likelihood tree with the lilRab 1 gene as an example, we find, oops, what happened now? All right. Wrong button. Sorry for that. No, come on. What's happened now? write these things?
Control L.
Control L, thanks. I tend to forget that. Yes. So this is now a maximum likelihood tree that we draw from the LILRA1 gene and including also other paralogous genes of LILRA1 in humans. We see that for the coding sequence alone, also LILRA1 groups outside of gorilla and chimpanzee. So just a brief information, what does LILRA do? It's a leukocyte immunoglobulin-like receptor of subfamily A. It's expressed in monocytes and B cells, and for that it plays a role in negative regulation of immune response. And it is a cell surface receptor linked which is linked to signal transduction. And maybe some of you recall the paper or the first true genetic difference in terms of a gene knocked out, for example, in humans that is active in chimpanzees and gorillas. There was a gene involved in also a cell surface antigen. So it's kind of interesting that we find here now a gene which favors the grouping of chimpanzees and gorilla, makes human kind of special, which is involved in immune response. And just to show you that this is not the only example in our data set, I show you here a list of human genes, just a partial list, where we find evidence for more than 300 base pair of exonic sequence that humans and chimpanzees are not their closest relatives. So what we have to learn from this is that a pretty high proportion of human genes in our genomes, so our estimate is about 1/3, contains at least parts that evolved human-specific already from the ancestral population shared by all three species, human, chimps, and gorillas. And now does that has anything to say? Well, we just now checked for amino acid substitution patterns. We just checked for two patterns. One is that human shares to the exclusion of chimpanzees as gorillas. So chimps and gorilla have the same amino acid and human has a different one. And the second pattern is where chimp has a different amino acid represented than humans and gorilla. And what we see is that in trees where human is grouped as outgroup, we find 59% of the total amount of this pattern represented, although these sequence trees make up only 14% of our amino acid coding sequence that we have analyzed. On the other hand, we see the very same picture for the chimpanzee, where chimpanzee has a different amino acid. We find that 41% of the patterns observed, or of the patterns observed in only 11% of the sequence. So it seems to be that the sequence genealogy somehow also influences the distribution of non-silent substitutions that might, of course, somehow are linked to the phenotype. Now, what I'm trying to convince you is that we now have to ask, by knowing that approximately 1/3 of our genes are evolutionary old, we have to, of course, ask the question, when did species-specific morphological characters arise? And that the naive assumption that people usually have, that just the presence of a shared derived evolutionary character in a fossil that is nowadays only seen by humans is actually not sufficient evidence, because due to the fact that morphological characters, of course, or the genealogy of morphological characters has, of course, to follow somehow the genealogy of the underlying genes, we have to assume from our data that Sahelanthropus, for example, which is actually the problematic taxon in our data set or in our fossils, might have actually been a fossil derived from an ancestor shared by humans and chimpanzees, but simply due to polymorphism in that ancestor population, shared an ancestor shares or displays a character which then, after the split of humans and chimpanzees, got fixed only on the human lineage, but not on the chimpanzee lineage. And the other possibility is, of course, that Sahelanthropus is actually part of the population shared by humans, chimps, and gorillas. So all this we have to take into account when we try to assign fossils to positions in that sequence tree. So now I come to the second part of my talk, and ask the question, what happens now when subsequent branches get compressed? So we now consider 4 species, or 4 populations, nonetheless, I will stay with species. And we have the case that not only T1, the time between T1 and T2 is compressed, but also the time between T2 and T3. So what happens if they are only sufficiently compressed? There's pretty high a probability that down here in the ancestral population shared by all 4 species, we have 4 separate genetic lineages now. And first of all, before I start talking about that in more detail, I ask the question, is that a scenario that biologists believe could be true? And there was a paper, 2005, by Rokas et al., and they claimed that there is a signature of radiation compressed in time in animal evolution. And what you see, what they did, they had, I think, a total of 50 genes which were nicely able to resolve the phylogeny of fungi, which, however, failed to resolve the phylogeny of metazoans, especially here in the early branching parts, although the evolutionary timescales are more or less the same. So we would expect if it's somehow informative, these genes are informative for that phylogeny, they should be also informative for that phylogeny. However, they are not, and what they concluded is that these speciation events, which gave rise to the early branching metazoans like sponges and cnidarians and whatsoever, that they occurred in a very, very short time scale. So we have to at least assume that radiation events where subsequent branches in the tree and the species tree get compressed are actually happened during evolution. So now, why I'm talking about that? The reason is because there is evidence, or it has been shown actually, that compression of subsequent branches can cause anomalous gene trees, or AGTs. It was shown by Dehnen and Rosenberg in a paper 2006 in PLOS Genetics. And what does it mean? An anomalous gene tree is an incongruent sequence tree that is actually more likely to be observed than any sequence tree that is congruent to the species tree. And just recall the situation for humans, chimps, and gorillas. There we could easily show that the most frequent sequence tree is actually always the species tree. Here, however, with 4 cases and 2 branches compressed in time, this is no longer necessarily true. So just to give you an idea why this is the case, the reason is that not all unlabeled rooted tree topologies, and rooted is now important, are equally probable. So consider again the case that you have 4 genetic lineages in a population. Any 2 of them will coalesce first, leaving us with 3. And then we have 2 possibilities. Either these 2 single branches will coalesce first, and then they will kind of form this symmetric topology of the sequence tree. And there's only one possibility to do it, so this will occur with in 1/3 of the cases. The other possibility is that either these 2 branches coalesce first, or these 2 branches coalesce first. And in 2/3 of the cases, then we will end up with an asymmetric topology. However, when we add now labels to these trees, the situation all of a sudden looks a little bit different. Because these symmetric sequence trees, there are only 3 possible labelings, right? Whereas for the asymmetric case, we have a total of 12 possible labels. So now, just a combinatorial probability now to observe any of these sequence trees is, of course, that any symmetric labeling will be picked with a frequency or with a probability of 1/9, while any labeled asymmetric topology will be picked or will be achieved with a probability of 1/18, simply by multiplying 1/3 the probability to be asymmetric, then by 1/3 to pick any of these topologies, label topologies. So what happens now if our species tree actually is asymmetric and the times are again sufficiently short that the majority of genetic lineages will not coalesce until prior to T3, then we will have the case that in 1/9 of the probability of a labeled symmetric tree will be 1/9, while the probability for an asymmetric tree, which actually is the original species tree is only 1 over 12, right? So if we condition that all coalescent events occurred prior to T3, then our probability for a symmetric sequence tree is 1 over 9, and by that larger than the asymmetric true topology. And by that we will have the problem that our sequence inference of the sequence of the species tree by means of the sequence tree, by the most frequent sequence tree, will be positively misleading. Now, do we observe something like this in real data? And again, now we go back to the human chimp gorilla orang example. In the first slides, or the first time I showed you this figure, I just referred to those that were informative for the human chimp gorilla grouping. Now we can consider those sequence trees which can be produced by a coalescent event down of the ancestor shared by human chimps, gorillas, and orangs. And what we see, in fact, is that the symmetric topology is observed more frequently than the asymmetric topology, as we kind of would expect it, actually. However, of course, this comparison, especially the numbers here, are a little bit unfair because a number of sequence trees I have not taken into account, since they are most likely generated by coalescent events prior to T3. Nevertheless, this is kind of a nice indication that the stochastic prediction somehow finds its reflection in the real data. So I've shown you the case for 4 taxon trees where only the asymmetric species tree can produce AGTs. How is it when we go farther into n taxon trees? Then here 5 taxon tree, the probability for each label tree is 1 over 90, 1 over 60, 1 over 80 in the 5 taxon case. And there exists a proof that now every species tree topology with 5 or more taxa, 5 or more species can give rise to AGTs. So it's more severe, actually, the problem than only with 4 taxa. And the conclusion is species tree reconstruction by means of taking the consensus from many sequence trees can be positively misleading. Now, the simple solution, because we somehow want to arrive with come up with a fast method to reconstruct species trees in the presence of AGTs. And our simple solution is that we simply take advantage of the case that rooted triples do not display the AGT phenomenon, right? And the reason is actually since any of these 3 species trees occur with sequence trees occur with the same frequency, so there is no topological factor, as I've explained it for the 4 and more taxon species trees. So our approach goes the following. We have a data matrix consisting of a number of taxa, of a number of genes. For each gene, we reconstruct sequence trees, so we come up with a collection of sequence trees, as in the human chimp gorilla orang case. Then we extract all rooted triples from these sequence tree collection. And from the rooted triples, now we get those that are congruent, that are the most frequent ones, right? Because, again, we think that the most frequent rooted triple, since there is no AGT, reflects actually the true species tree. And from these collection now of rooted triples, we then use consensus methods to come up with a consensus tree. And what we use in our case is actually the quartet puzzle algorithm in a slightly modified version. And we call it triple consensus method, or TCM. So, of course, we did some simulations if that really works. What we did was we generated a species tree with a Yule prior. According to the species tree, we used the coalescent model to generate sequence trees under different theta values for the theta parameter, for theta. Then we chose 2 ways. One way was we took the sequence tree as it was simulated, so we excluded any sequence tree reconstruction error. Extract the rooted triples, get the most frequent ones for each triple of species, and reconstructed the tree. And as a second approach, we used these sequence trees then to simulate short alignments, length of 200 base pairs, use maximum likelihood tree reconstruction to reconstruct the tree, and then joined followed with the same procedure as path A. So the results for that simulation are actually shown here. We have, as I mentioned, a number of values for theta, where a high theta corresponds to a higher probability for AGTs to occur. We have two methods for getting the species tree, to infer the species tree from the sequence trees. First of all, our triple consensus method, and then the extended majority rule method. And what we see now is that for different numbers of genes, we get different accuracies depending on the theta values and the chosen method. And accuracy means that the topology of the species tree that we reconstruct is identical to the simulated one. And just to give you an idea, we chose 20 taxon trees, and for each parameter combination, we made 1,000 replications, and the numbers are actually just the percentage we recover the true species tree. And what we see is that for low theta values, actually both methods pretty much behave the very same. With a few genes, only 10 loci available, we have quite a bit of error in our tree reconstruction, simply because we are not able to assess the sequence trees. And the more sequence trees then we consider in our analysis, the more accurate actually our estimate gets. Good. Now, however, when we increase the theta values, for example, we see then that for 0.1, for example, theta 0.1, we end up with a very, very low accuracy for low numbers of genes analyzed. However, when we now, for example, look at 2,000 genes, we recover the correct species tree in 82% with the TCM method, but only in 61% with the MRE method, which actually has to be due to AGTs occurring in our simulation. And of course, that gets more severe the higher the theta gets. For theta of 0.5, we end up with MRE being of no use at all, whereas the TCM, when we have sufficient numbers of genes, and this is of course sometimes the problem in real data, we will still quite likely reconstruct the correct species tree. Now, of course, we have to consider the effect of errors in sequence tree reconstruction, and this is what we did here. So this is now the path B I've showed you initially, where we did maximum likelihood tree reconstruction, and down here, just for a comparison, where the case where we have the true sequence tree topology given. And what we see that for low numbers of sequence trees, we have quite a bit of a problem. So we perform way worse than when we know the true sequence tree. However, the performance recovers with increasing numbers of sequence trees available. And what we also see is that both methods, MRE and TCM, are affected approximately to the same extent by errors in sequence tree reconstruction as we actually would have assumed. And for that reason, we conclude that sequence tree reconstruction errors are simply adding kind of general noise to the whole thing. Now, how is it with application to real data, to real biological data? It's of course interesting whether this is now just a made-up scenario or whether this has real implications in real species tree reconstruction. And for that reason, we went to the InParanoid database. For those who don't know it, it's a database which assigns orthology relations between pairwise orthology relations between two species, and they use only completely sequenced genomes or proteomes, right? And I will not go into detail of that approach, how they assign pairwise orthology. The only thing I would like to show you is how we then, from this database of pairwise orthologs, then ended up in a set of sequences that are orthologous over all 20 taxa, which we considered from that database. And what we did was we started with a human protein. Here we identified its orthologous protein in Cyona, and then we took this identified Cyona ortholog, checked for the ortholog Anopheles, and then hopped from species to species over all 20 species until we ended up with a chimpanzee yeast ortholog pair. And then we took the yeast ortholog and identified again the orthologous protein to this in humans. And then we asked, is that the very same protein we started off our trip? And if yes, we included it in our data set. If not, we simply discarded it. And by that, we ended up with a set of 216 genes. We had evidence for orthology, and this, of course, we would like to have. And we used now both methods to reconstruct the species tree. And this is actually the species tree we ended up with. The numbers are the number of sequence trees in support of these clades. And first of all, it's a pretty good species tree, so it recovers what people usually believe that is the sequence tree. However, there is no difference between the tree obtained by MRE, and for that reason, there is no indication for AGTs in this data set. Does that now imply that we can skip AGTs at all, the idea of AGTs at all? Presumably not, because there might be more relevant data sets. And now again, coming back to the data set of Rokas et al., you recall that they have these early branching metazoans in their data set, which we actually didn't have. So there might be actually more relevant data sets. And this tree I show you here is the outcome, the first outcome of the priority program of deep metazoan phylogeny in Germany, funded by the DFG, where we are participating in. And this is based solely on EST sequence data, right? With the exception of 5 species: yeast, humans, C. elegans, Anopheles, and Seon, I think. And here we see now that we first of all have also the early branching metazoans represented by a couple of, I think, 8 sponge species, and also other very deeply branching groups. And what we also see is that the internal nodes here from that maximum likelihood tree are pretty short. So this actually might be a more relevant data set to check for the influence of AGTs. However, the problem is that this is still a very patchy data set, and this is due to the EST sequences, which are actually not evenly distributed for all genes, for all taxa in that species tree. And for that reason, we have to wait a little bit until we hopefully are able to complete the data matrix for this taxon set, and then we might again recheck whether AGTs play a role or not. Now, coming to the end of my talk, there are some conclusions and challenges. And first of all, I mean, the biggest challenge, I think, I don't have to mention, and this is actually the that we have to somehow, first of all, understand that the genealogy of the phenotype essentially depends on the genealogy of the underlying genotype. So we have to consider population genetics effects especially when branches get short or the population sizes get large. And this actually brings me to the foraminifera talk, where I was simply wondering how large is the effective population size of the foraminifera, right? So this is actually one of the most straightforward things to conclude, but I think also one of the biggest challenges, because many people will have a very hard time to accept this. So then, of course, for understanding human genetic ancestry, and especially human evolution, and also correctly interpret the fossil record, we kind of need a map of the human genetic ancestry. And this essentially depends on the availability of the data, firsthand, and second, of course, on the possibility to accurately reconstruct the phylogeny, although there are very few informative sites. Then there's as a conclusion that these ancestral polymorphisms and incomplete lineage sorting serve as a very nice explanation for homoplasy. So whenever you have a set of species and you find evidences for homoplasy in the morphological characters, it might be actually due that this is not convergent evolution, as people usually put it, but this is simply due to actually incomplete lineage sorting of ancestral polymorphisms. And cichlid fishes, I think, are a very nice example for that. And eventually, of course, that short branches can lead kind of to an unintuitive behavior of evolution and can in several ways interfere with the accurate species reconstruction. And this is not only AGTs, but also due to the phenomenon of long branch attraction. So I think there are sufficient challenges just in mean in terms of where we need to think. It's not challenges that I can point out, do this and do that. Just maybe it's a way of how we should, in future, a little bit try to think when we really want to understand evolution. And the acknowledgements, as I mentioned, the work I was presenting is work of a total of 5 people. We are at the CBIF, and we got funded by the Viennese, I don't know how you call it in English, Wiener Wissenschafts- und Technologiefonds, also funded by the DFG, and I forgot BIN2, the Bioinformatics Network, which also, I think, funds Greg. And thank you for your attention.
So you constructed like 215 trees or one tree. How you discarded the possibility of lateral gene transfer?
So first of all, we didn't pay attention to lateral gene transfer at all, because usually in the species we are looking at, so these were metazoans, with yeast as an outgroup, there is no clear evidence for lateral gene transfer to occur. And second, lateral gene transfer will influence certainly only single genes, right? But not the majority of these. So they might, even though lateral gene transfer might exist, which of course I would never argue against, because it could be, right? It, I think, wouldn't influence the outcome of our analysis. It would be simply leading in some gene trees supporting a different grouping, which actually we'll, of course, see. Yeah.
How does the uncertainty of coalescent times increase as you go further back in time? And does that affect the possible importance of these processes for deep metazoan branches?
So the coalescent processes actually do not affect this problem at all, because I mean the coalescences we have now in the present species, you would assume all of these are actually finished until the species reaches the next common ancestor. So there are no genetic lineages available today in one population that go back down to the root of metazoans, right? So you have, even at the root of metazoans, you have only the lineage descending then down to the, which then coalesce in this ancestor population. So I don't think that the increase in error that you get in the coalescent process will kind of interfere with it, with the reconstruction. So I don't think that it plays a big role.
I didn't quite get, you made this basic point that, that so long as the branch length is non-zero,
yep,
there will be some coalescent events on it,
yep,
and that means it tips it in favour of trees that do support the species
right.
tree. And then, but then I didn't get your point about, you know, you condition on a certain history up to a point, and in that way you get this bias towards the wrong tree or the
Yes.
wrong pass of tree.
Yes.
So, how do you know how do you have that data to do that conditioning?
So the conditioning is simply the explanation why in a star topology, for example, or when the internal branches approximate zero, why we end up with the wrong conclusion, right? So even if they are non-zero, but closely enough to zero, because if they are non-zero, we have a resolved species tree, but we might not have any coalescent event in there, because the time is too short, right?
So we're exposed to stochastic fluctuations.
Right.
I see. So then I expected you to come back and explain to us, you know, because you had that mismatch between, you know, between the different calibration points.
Yep.
And I thought you might come back and say, well, we could get an idea of exactly how big the overlap is. You know, how far past the how far past this point we can go, and it is past the species coalescent point we can go by using the genetic coalescent, and to see whether that time was enough to make up what you needed with the mismatch in the calibration, because it gives you an idea of the population sizes.
Oh yeah, so there are of course ideas about population sizes, and also we estimated them, and other people estimated them as well. But it's a very tricky thing, and the reason why I didn't go into detail with respect to the fossils is that we have absolutely no idea in what time scales morphological characters arise. So I personally have no idea whether it takes 1 million years to evolve to an upright position, or whether this goes way quicker. So this would be just pushing the thing a little bit too far, I would say. So for me, the whole issue is also, there might be alternative explanations. For example, Patterson, they kind of had a different scenario where they said that humans and chimpanzees split initially something like 7 million years ago, and then they evolved as separate species, so giving rise to species-specific phenotypes, and then like the Sahelanthropus, and then they rehybridized in order to explain the young genetic age, which is also kind of an explanation. And they say we've seen something like rehybridization in animals before, and yeah, sure, it's one possibility. However, you cannot exclude the scenario that I have drawn, right? And this is actually, we need to think, and the argument, our strong argument is that it's really a huge number of genes, where at least parts kind of really have a very, very deep ancestry. So that we need to consider these evolutionary scenarios, these morphological scenarios that we have drawn. But it's not the all-embracing answer, right? Homoplasy will exist. Convergent evolution will exist. But we have to consider if it's taking too much homoplasy events, then I think one could think, oh, might be that actually speciation times are pretty short. And the reason why I'm talking about this was that I heard a talk by Axel Meyer, and he was talking about cichlid fishes, whereas we know that speciation events occur very, very rapidly in the African lakes. And he was happily talking about convergent phenotypes in species that are not their next closest relatives, where you see the ancestral phenotype in actually really the closest relative. So you have apparently homoplasy, and this occurred very, very often. I was simply asking, or was wondering, could that be true, or couldn't that be simply lineage sorting and ancestral polymorphisms? And this is just the way of, I would like people to think. Just consider a different evolutionary scenario.
No, okay, then I think we thank the speaker again.